Okay. So, hello everyone. Thanks for having me. I would like to use my presentation essentially to tell you a brief story regarding the acquisitions of resistance to targeted therapies in lung cancer and highlighting the importance of understanding what drives this resistance. So I figure if I'm telling you a story, I might as well start at the very top. And data shows us that lung cancer is leading in terms of diagnosis and in cancer-related deaths worldwide. If I were to take the time to dissect these values and explain the categorizations of types of lung cancer being diagnosed in terms of histological classifications or oncogenic driver mutations, we could understand what is being diagnosed and where. But I'm not doing that. What I'm going to say is that for this presentation, I'm mainly going to focus on EGFR-driven adenocarcinomas. And the thing about EGFR-driven carcinomas is that EGFR, it is a targetable mutation. So if a mutation is detected in EGFR gene, a specific therapy can be employed to it using tyrosine kinase inhibitors that eliminate cells that specifically carry that mutation. Hence, we get targeted therapies. And as we know, in a broad way, these targeted therapies offer a better outcome in therapy for patients, which is fantastic. However, this brings us to the big antagonist of this story that I'm telling you, which is resistance, because the vast majority of the patients treated this way become resistant to the therapy in about a year. And in a very canonical and classical way to look at resistance, development in this case would be to say that initially there's a mutation, that the cells carrying the mutation expand, eventually the patient has symptoms and gets diagnosed, treatment starts, and we get the elimination of cells carrying this sensitizing mutation to that therapy. However, along the way, a resistance mutation to that specific therapy can arise and get selected and become the bulk of the tumor, and now the tumor is resistant. But we also know that this is a very incomplete image of what happens. And what we find in the lab is information regarding this in order to add more information. So if we were to look here first, this essentially shows us what the last image shows us, where a therapy-sensitive mutation gets detected, and therefore erlotinib treatment can start, and we have a response from the patient with elimination of the cells, and everything is very stable for a while. But eventually, a resistant mutation, represented by these red bars, occurs. And what we see is not only this happens, as I said in the previous image, but there's always also the resurfacing of cells carrying the sensitizing mutation. So these cells that initially were sensitive to therapy, along with these cells, are growing, and none of them are responding to the initial therapy. And only after employing a third-generation tyrosine kinase inhibitor, or osimertinib, we get the elimination of the cells. If we look then not at one patient, but at a lot more, we can see that if we compare the allelic frequency of resistant mutations and sensitizing mutations within these patients, the allelic frequency of the sensitizing mutations is almost always higher than the resistant ones, showing us that in the resistant tumor, only a fraction of cells actually carry the resistant mutation, and the others only carry the sensitizing mutation. Yet, all of these cells survive. So this information led us to essentially create a hypothesis regarding this. And what we're saying is that, yes, okay, so vertical transmission, where a cell carrying a resistant mutation passes down that mutation to daughter cells and proliferates, occurs, but there also appears to be an horizontal transmission or mutation of resistance, where these resistant cells are able to communicate with their environment and confer resistance to sensitive cells without them ever acquiring a resistance mutation. And this will allow us to explain the fast dissemination of the resistance phenotype within the tumor, as well as the stability of this resistance. So having got here, we wanted to test this in the lab to see if we can evaluate and get information regarding this communication that is occurring. And to do that, we essentially developed two experimental models, one in vivo, one in vitro. And they both kind of rely on the employment of cells carrying a sensitive mutation and cells carrying a resistant one. And the in vitro one, it's a very relatively simple experiment where we plate sensitive cells and we treat with increasing concentrations of erlotinib, Or we do the exact same thing, but we also supplement the cells with conditioned medium from the resistant cells grown separately. And what we found is that even though both these experimental groups acquire resistance eventually, the conditioned media treated cells acquire resistance much faster than the other ones. Indeed, there's in fact a whole month difference between these two groups. And we checked, and there's no contamination from resistant cells within our cell dish, so these are the initial cells just modified somehow. And if we jump to the in vivo model, it works very similarly, where we have single inoculated and dual inoculated mice. And essentially, in the single inoculated mice, we only inoculate with sensitive cells, we treat, and we wait for resistance, while in the dual inoculated ones, we inoculate in one flank with sensitive cells, and the other one with the resistant cells, and then we do treatment. And again, we found that the presence of resistant cells hastens the speed in which resistance occurs, and again, with no contamination from our cells in our sensitive tumor. So this led us to a lot of questions, but if I were to group the questions in two main categories, I would say that we were at this point interested in knowing what and how the resistant cells are disseminating, let's call it, this resistance, and how are sensitive cells being reprogrammed by this communication. For the sake of this presentation, I'm focusing only on how these cells are changing. And to study this, we essentially conducted a comparison-driven approach when we had a lot of samples from the tumors of the mice that I talked about just now, and we wanted to see what changed before and after resistance acquisition occurred. So that's what we did, and what we found is that, indeed, there is a change in the transcriptional landscape of these tumors after resistance acquisition. And at the very top of this list, we found endocytosis, which for us was very interesting because we were looking, this tells us that these cells appear to be receiving more information or being able to process more information and receive them from resistant cells. And when we look at endocytosis, we found that the top most expressed genes here were caveolin 1 and 2. Now, these genes code for proteins that are able to conduct clathrin-independent endocytosis, and they're able to modulate the shape of the cellular membrane in order to create kind of a communication hub where signaling molecules get concentrated in order to be near membrane receptors, which again was interesting to us. And after validating this in mouse tumors, we went and see if it also happens in our patients. So we had a cohort of patients that had a biopsy at diagnosis and upon disease progression. And what we found is that, yes, so caveolin 1 and 2 are more highly expressed after disease progression than they were in the initial tumors. So following the same line of comparison, we're looking at transcriptomics, we then looked at proteomics and phosphoproteomics to look for proteins expression and protein activations that differ after resistant acquisition from the beginning of the tumor onset. And we found part of our focus was dedicated to proteins associated with cell proliferation, cell survival, anti-apoptotic signaling. And one I'm highlighting here today, that will bring us to the last result that I'm showing you, would be PI3K, which I think we can all agree fits the bill when it comes to pro-survival, pro-proliferation protein. And the question that we asked is, okay, so what happens if we inhibit PI3K expression? What happens to the cell's response to treatment? And to do that, we use two cell lines. So we use the initial sensitive cell line, the parental one that is sensitive to erlotinib treatment, and we use the cell line that had acquired resistance through conditioned media exposure. And as we see here in the first result with the sensitive cell line, when using the PI3K inhibitor vortmannin, we see that whether we are using the inhibitor, okay, so stop working, or the DMSO in the gray bars, we don't see overall a big difference when it comes to the response the cells have to erlotinib treatment. In fact, if we focus individually on what each one does, DMSO treatment of the cells doesn't really change at all the way that they respond to erlotinib, and so does not vortmannin treatment to the cells. They keep dying, they're sensitive, and when we compare the both conditions with erlotinib treatment, there is no statistical significant differences there because, long story short, inhibiting PI3K here does not change the way that the sensitive cells respond to therapy. However, when we come to the resistant cells, the picture changes a little bit, right? So we see that it appears to be a difference in treating the cells with a PI3K inhibitor or just not treating them, right? So if we focus on the individual results, when the cells are previously treated with DMSO before erlotinib treatment, there is no significant at all reduction in cell viability. The cells are resistant, they keep being resistant, this doesn't change anything. But when we treat them with PI3K inhibitor, we find that the cells start dying now. The cells become sensitive, somewhat sensitive to the erlotinib treatment. And when comparing the two groups again, with erlotinib looking at the differences between treating and not with PI3K inhibitor, we see that now there is a difference, and by inhibiting PI3K, we resensitize the cells somewhat to the initial erlotinib treatment. So overall, these results, what I wanted to do with this presentation and with this result itself, was to highlight the importance in understanding what is driving resistance acquisition, in this case through interstellar communication and alterations in cell and expression within the cells, in order to be able to adapt and understand this cell, the way that overcome resistance in therapy. I would like to just use the last slide to thank everybody at I3S, all the research group, as well as the scientific platforms, and all of you for your attention.